Good afternoon. Welcome to the UCSF Academic Senate's 66th Annual Faculty Research Lectureship in Basic Science. I'm Kartika Pallar, Associate Professor and Chair for the UCSF Academic Senate Committee on Research. It is my honor to introduce the Academic Senate Faculty Research Lecture. The Academic Senate's Faculty Research Lectures started in 1957. The first lecture was given by Professor Melvin Calvin on the topic of photosynthesis as part of the Charter Day activities. Originally, an ad hoc committee was appointed to nominate the first faculty research lecturer. Then from 1960 on, the Committee on Research decided to take on this responsibility, and in 1966 decided to solicit new nominations each year from the deans, department chairs, directors of institutes, and members of the Senate. Since then, nominations are made by UCSF faculty who consider the scientific research contributions of their colleagues and submit nominations for this prestigious award to the Academic Senate Committee on Research. I'd like to thank the committee for their hard work. This Faculty Research Lecture Award recognizes exceptional career achievements in basic science research. And today we are very proud to honor our colleague, Dr. Andrej Sali. Before we begin, a few housekeeping points. There will be time at the end of the program for questions and answers. For those joining us by Zoom, please submit your questions and comments using the Q&A function at the bottom of the Zoom window. And this event is being recorded and will be available on the Senate website. Now I am pleased to present Dr. James Fraser, who is the nominator of Dr. Sali. He is a professor and chair of the Department of Bioengineering and Therapeutic Sciences at the UCSF School of Pharmacy, and will give us an introduction to Dr. Sali. Dr. Fraser.
Well, thank you so much, and thank you to the Faculty Senate for organizing such a wonderful series of lectures from year to year, and this year in particular. It's such an honor to introduce our beloved colleague, Andrej Sali. So I was looking through for the oldest picture that I have of me and Andrej together, which is us looking very upset outside of, I believe, Tanya Cortemi's office as we tried to induce her to drink some wine or something one night. And then you can see, Andrej looks basically the same about 15 years later, but I've aged significantly, during that time. Andrej's been, during that time I've known him, Andrej's been just an amazing colleague and mentor. But there is one complaint I have about Andrej, which is that, my friend Nevin Krogan, who sends his best wishes and can't be here today, and I noticed a disturbing pattern in Andrej's talks, which is that Andrej really loves to point out that everything in all of biology can be explained only through 4 stages of modeling. And Nevin and I started taking pictures of Andrej explaining the 4 stages anytime we would see him give a talk. And so here you can see he's actually in Berlin giving a talk, and that's a photo by Nevin. And at the exact same moment, I am texting Nevin a photo of Andrej giving the 4 stages. And those, 4 stages are really important and will be really foundational to the rest of this intro, and I'm sure Andrej's talk. But I'd just like to point out that this is something that we've really seen a lot of through the years. Andrej is a big fan of these 4 stages, and Nevin and I just keep noticing that Andrej uses these slides with the 4 stages over and over again. And we really enjoy taking pictures of Andrej. He's not aware of this until now, delivering the 4 stages all around the world throughout UCSF. And I'd be remiss if I didn't point out this morning, I was in Andrej's office for a thesis committee meeting, and I took this picture where you can see his student Matt, who's not there, also has 4 stages on the slide as well. And Matt Jacobson is there, of course, watching. And so, I just wanted to bring you through the 4 stages of Andrej as I've come to know him and appreciate him. And I start with the first stage, which is gathering data. So Andrej, as you will learn if you Google him, is Slovenian. Sali actually may be derived from Solomon, which is my middle name. So we share that in common. But he was born in it was former Yugoslavia, now Slovenia, which is, of course, most famous for now Luka Doncic and Melania Trump. But Andrej ranks somewhere high in the list of famous Slovenians. The next step of modeling is spatial restraints. And so as we see where Andrej extended spatially, he moved on as a graduate student to England, working with Tom Blundell, where he published a paper that is, so influential in all of the exciting activities in AI as applied to biology that, really I can't properly encapsulate it. But this paper on comparative protein modeling by satisfaction of spatial restraints is, his highest cited paper. And reading it last night again, I am just like awestruck at how profound it is and how much foresight was contained in it. Andrej always has a way with words. Even this morning when we were in this thesis committee meeting, the way he uses the word satisfaction is always tinged with a hint of mischief that I think stems from his musical fandom. And I think that the title here is really an homage to the Rolling Stones as well. And then after his amazing work with Sir Tom Blundell, he moved as a postdoc to the United States to work with future Nobel laureate Martin Karplus, where he worked a lot on very theoretical models of protein folding. And for those of us who've ever been in a qualifying exam with Andrej, we will know that his pet peeve is the word understanding. If a student says, I hope to understand a certain phenomena, Andrej will nail them. What does that mean to you? What does understanding really mean? So I found it really amusing to find one of his postdoc papers actually titled "Understanding Protein Folding" by free energy surfaces from theory and experiment. All right, so after these spatial restraints comes configurational sampling. So Andrej began his configurational sampling in the Upper East Side in New York at Rockefeller University. This was the age of the beginning of structural genomics. And this is also where Andrej really started integrating in different types of experimental information into comparative modeling and de novo modeling. And I think the first paper where he really did that sort of looks at these histones from various organisms and how to model them relative to each other. And it was there that he started working on the nuclear pore complex. And fortunately for us, we were able to recruit him here to UCSF about a little over 20 years ago now, where he has just continued to provide not only foundational work on the theoretical aspects of modeling, but also just amazing, breathtaking applications to a wide array of biological systems, most notably the nuclear pore complex, which is the subject of just several landmark papers from Andrej's group that I hope he will touch on today. Yes. The fourth step in the fourth stage is validation. And this is easy to do for Andrej. He's been validated by many awards, including election into the National Academy. He's been a founder of multiple successful biotech companies. And, he's an amazing community citizen. His time as a QB3 director was notable for many things, including the recruitment of just amazing QB3 fellows. But he also led the Winton cluster for quite some time, was a community member leader in the journal Structure, and just has been an amazing mentor and friend and colleague to all of us. And as all the students know, Andrej is also just a very proud Bayesian. But I think the most important part of the validation step is like, hey, that's kind of what we're doing here today, is validating Andrej. So, please join me in recognizing the 4-stage development of this amazing colleague. And let's end with a hearty round of validation for Andrej Sali.
That's quite amazing, actually. I swear to God, I haven't shared my slides with Jamie. Maybe we're done. Okay, well, thank you so much for this honor. It comes with a lot of stage fright, actually. Maybe my first talk in English was a little more difficult, but this comes next to it. I'd like to start by acknowledging my friends, like Jamie, who nominated me, and collaborators and students that this recognition really applies to primarily, not even just so much me. And so let's get going. A few comments on the title. Good synonyms for harmony in this title are consistency and minimal frustration. And I'd like to tell you my story about how my perspective on modeling evolved from the student days until today, with a quick peek, hopefully, into the future as well at the end. And I'll try to convince you that harmony is a key concept when it comes to modeling. Modeling creates harmony among various aspects of a model that it produces, between the model and the data on which the model is based. And perhaps most deeply, it also creates harmony among the data themselves. And then, maybe even sometimes among the scientists, including experimentalists and modelers. Jim, good guess. So let me make a few very generic comments on modeling to begin with. The context in which it appears and what it tries to accomplish in science. As you know, science really aims to make predictions and explanations about natural systems, and it does so by iterating through experiment and modeling. And modeling in particular aims to convert some input information about the system of interest, including importantly experimental data about it, into a model that's hopefully more useful for making testable predictions and explanations than the input information itself, on which the model is based. So the part of science that actually makes predictions and explanations is the models, not the data. And then given this role of models in science, there are some model qualities that are worth highlighting that we'd like to maximize or at least optimize. We would like to compute a model objectively, automatically, and reproducibly. And we'd like this model to be accurate, precise, complete, and optimally simple. And these panels at the bottom hopefully very quickly convey, at least approximately, what I mean by these attributes. And this is true for any kind of modeling, I think, and including for the type of modeling we wish to practice, we wish to develop, except that, of course, we do it a little bit more specifically in the context of structural biology. And so this slide kind of defines the scope of our research in our group. Over many years, structural biologists have determined structures of many biomolecular systems. These systems, target systems, are becoming increasingly large, complex, and dynamic. They consist of multiple, even hundreds of protein subunits. The first atomic structure of a protein was determined in the 50s, Perutz and Kendrew, by X-ray crystallography. But today's systems, target systems, are much more complex. And we can be addressing spatiotemporal models of cellular neighborhoods, and increasingly progressing towards targeting an entire cell at near atomic level of detail. But of course, traditional methods, such as X-ray crystallography and even cryo-electron microscopy, are not sufficient on their own to achieve these goals, because these are too complex systems to describe with single methods. And so our group is developing applying and disseminating, importantly, modeling methods that can expand the scope of structural biology. And we do so in two ways. We try to expand the types of experimental data that one can include in computing these spatial temporal models. And second, we are expanding the types of models that are computed based on this data in order to contribute towards expanding the scope of structural biology in the context of cell biology. So that's our goal for today. And what I'd like to tell you is how we got to this point, basically mimicking Jamie's introduction. Just since I have more time, hopefully, it will be a little more detail. But thank you, Jamie. I mean, it's amazing. You would have saved me a lot of time if you told me about that construct some time ago. But basically, that's how I constructed the talk. And as Jamie indicated, I come originally from Kranj, a city in Slovenia, which a long time ago was part of Yugoslavia, and that's how it looks. I have a younger brother. My dad was a professor of psychology. He was into dyslexic children research. My mother was a mom. My first computer in primary school was this Hewlett-Packard calculator, which was programmable. It had 150 bytes of memory. My first program was solving quadratic equations. And I do want to say that I was very influenced by a biology textbook in high school, which I remember so vividly was a result of an initiative in the US by Vannevar Bush, who was a president, an advisor, a scientific advisor to multiple American presidents, who initiated this program, spread all over the world, including to Yugoslavia. And I became excited. That was my US connection right there. I was also very fortunate to be able to convince my first mentors at the local Premier Institute in Biochemistry, Institute Josef Stefan, to let me do experiments in the lab as a high school student. And I shout out to Vito Turk, who was the founder and the head of that department, who was my first mentor, together with a few others. As it happens, Boris Turk, his son, just happens to be here today, staying with me for a couple of days. He's a preeminent scientist in his own right. I upgraded my computers. I had a personal computer, an Apple II Plus clone, shown here, which had to be smuggled by my dad and me from Germany to Yugoslavia. It was illegal to have personal computers in Yugoslavia in those days. We almost got caught at the border. Separate story, but it certainly changed my life, I can say. And I attended University of Ljubljana doing chemistry research there. Okay. Boris can tell you some stories later too, perhaps. So here's a sampling of two papers from high school undergraduate days. The first one was actually about purifying proteins from various sources, some of which maybe I'll avoid mentioning exactly, but I was doing some wet lab work. The gels look much better than this photo indicates here. And then I was very interested, mostly interested in computational analysis of protein sequences and structures. And this is a paper that tried to reconstruct the evolutionary, the evolution of a set of protein sequences, cysteine protease inhibitors, based on comparing the sequences, but also there's predicted secondary structures, which I did with my own program. I did some enzyme kinetics fitting, some CD spectra fitting, and things of that nature in high school undergraduate days in Slovenia. Then I was lucky again. Professor Took introduced me to Tom Blundell. And after a few visits, short visits, as an undergrad in London, I became a full-time student, graduate student at Birkbeck College. And I wanted to really focus on computational structural biology and just do it as best as I can. That was basically my goal. It's very kind of simple to state. And my first project, computational project, after doing a few X-ray structures of aspartic proteases, not difficult studies because there were established protocols, was this problem of how to compare dissimilar but still related proteins when sequences are difficult to align and structures are difficult to superpose. These problems may be easy in cases illustrated here in the panel, but when the sequences are dissimilar and structures are different, it's not so easy. And so the solution was simple. Let's just use both sequence and structure information, and let's use different kinds of sequence and structure information, positions in space, hydrogen bonding networks, the hedral angles, secondary structure states, and so on, to increase the amount of information that goes into optimizing a degree of similarity between compared proteins according to the alignment that had to be searched for an optimal alignment as part of this comparison. And so this maybe is the first example, a small one, of harmony that will keep up being more and more prominent in this talk, the alignment that we had to create through computation, in a sense, was achieved by maximizing harmony among compared proteins based on some scoring function that reflected the degree of similarity in terms of sequence and structure attributes of the compared proteins. There was then quite smooth transition from this comparison problem to the next major project during my PhD, which was to develop a method for more accurate comparative protein structure modeling. In comparative protein structure modeling, we wish to predict a structure of the sequence, a member of the family that has other members of the family of known structure. And so that they provide a good starting point for comparative protein structure prediction. And the idea and the problem was to do it better than just copying big parts of known related structures using their alignments to the target sequence to get the model of this target sequence. Not do that, but do it better by realizing that the comparison and modeling are in fact related. You just exchange what's known and unknown. And so the idea was to start with the alignment now of the sequences, target sequence with one or more known structure at the top, and then extract spatial restraints on the unknown, on the sequence of unknown structure, given its alignment with one or more known structures, using statistical preferences that were empirically observed, relating sequence and structure similarities for proteins of known structure compared with each other, supplement these homology-derived restraints with stereochemical restraints, bond lengths, the hedral angles, atoms don't overlap, and so on, and then obtain a model, compute a model by satisfying all of these spatial restraints as well as you can. And that's illustrated on this plot on the right. And the resulting program modeler became over time quite successful in terms of other people using it, is indicated by 200,000 or so academic licenses so far. It was also licensed to a company called Molecular Simulations a little later that disseminated it to biotech companies, pharma companies, where it was used for comparative modeling quite extensively. Of course, now with AlphaFold and other deep learning methods, it's finally time for this program to get retired, I suppose. And there were still, of course, problems with comparative modeling. The accuracy might have been insufficient of the models that it produced. You didn't always have a template structure. So already in 1990, based on the early work on Modeler, we proposed in an opinion piece in TIPS that to overcome these problems, one could use more information to apply more spatial restraints on the sequence whose structure needs to be modeled, and therefore get a more accurate or model in more cases. And there's a funny story here, because I wrote an essay in 2021 in recognition of the 50th year of the Protein Data Bank, in which I discussed integrity modeling, and I didn't cite this early review, of which Tom was a co-author, of course, senior author. And a reviewer pointed it out that I missed a major, and so the joke was on me. The reviewer was, of course, Tom. But it's true. It's an idea of it's an example of how a generalization is good to apply, even if only conceptually, when you develop computational methods. We haven't done anything about it at the time, but we still published a paper, an essay on it. And still, it was good enough for me to actually write my postdoc fellowship proposal on that topic. That's what I proposed, to do integrative modeling for my postdoc with Martin Karplus, who is, of course, a very impressive person. He trained, even at the time when I was considering his lab, he was a major force in the field. So many computational biologists, structural biologists were trained by him, and that was very attractive. Also, he represented a very different area of expertise, that of a physical, statistical, mechanical perspective on proteins, as opposed to a database, statistical or bioinformatics perspective. And I wanted to learn from that, and that's why I joined his lab. But as fate would have it, he completely ignored the fellowship proposal on which we agreed, and asked me to address a different question, which is, what's the difference between folding and non-folding protein sequences? And he also suggested, together with Evgen Shapnovich, to address this question using a very simple lattice model of protein folding that's indicated on this panel. The study started by creating a large number, hundreds of sequences within this model, for which we knew what the native states, lowest energy states were, simply by enumeration. And then empirically, with a Monte Carlo procedure, test which ones of them fold and which ones don't, which ones find at native states and which ones don't. And once we had these samples computationally generated, we just needed to compare them, folding and non-folding sequences, in various ways. That's where some creativity came in. And we discovered that a necessary and sufficient condition for folding in this model was that the native state be pronounced global minimum on the potential energy surface. And this was a nice result. It was noticed. Buzz Baldwin wrote a very generous news and views accompanying our paper in Nature, but then that had some negative consequences. There were a number of other scientists who felt they also deserved more generous comments than they received, and this is my atonement. I in these three panels on the right, I highlight three of those related bodies of work, and they will also serve for me to harp on harmony again. Nobuhiro Go, much earlier, proposed what's called the Go model, where he was studying protein folding with potentials that had terms favoring native contacts. And he called that the harmony principle of protein folding. The native structure of the entire protein was consistent with the local structure as reflected in those local favoring potentials. There were Joe Bringstone, Peter Wollness, who developed basically an analytical model of random energy model of polymer folding, where they observed that by minimizing the frustration on the energy landscape, you favor folding kinetics to a stable native state. And of course, our own here at UCSF, Ken Dill, has done a lot of work on protein folding funnel as well, along similar lines. And so all of this have this key concept about the landscapes that have the right features facilitating protein folding that can be described as manifesting harmony between local structures, local restraints, and the overall structure. What's an interesting joke here, maybe a little bit, is that one and the same thing was called harmony by Nobuhiro Go, East, and Peter Walliness called it, instead of maximizing harmony, minimizing frustration, West. But it's the same thing. And so after my postdoc, I again was fortunate that John Currian and Stephen Burley were looking for a computational biologist to recruit to Rockefeller University as a faculty member, young faculty member. And that's where I moved. And I decided not to continue focusing on protein folding, but to go back to my roots, so to speak, a little bit, and continue working on comparative modeling and integrity modeling instead. And although we're improving the basic methodology of comparative modeling, including model building and model assessment, I would only like to highlight one aspect here, and that's automation, large-scale computation, and dissemination of these comparative modeling tools. Because it is this that led to 3 other developments. First, helping formulating structural genomics, founding of companies, and pivoting full-time into integrative structure modeling of large complexes. But it kind of all naturally followed from this basically tool method development that I am highlighting here. So we automated modeler, comparative modeling. We could do it automatically on a large scale. This is the flow chart. We constructed the data. We ran that pipeline on all known protein sequences at the time. We created the database of these models, and we assessed these models. And so we served models and assessments and the web server, the tools, to everyone who cared as much as we could. One of the spin-offs was our contribution to formulating structural genomics. And I think I will become mindful of time. So you know what, I'll just say that that was one of the spin-offs. I will not belabor the point. The second spin-off, which I don't want to skip, was a discovery that it's really good to share as much as you can everything that you produce, especially software. But in order to do so, you have to make software robust, automated, documented, open source, and also you have to demonstrate it by applying it. And so in '94 already, Modeler itself was licensed, and with generous support from Tom Blundell and Martin Kaplas. They didn't want anything in return. Maybe they didn't know how much it would sell, but I'm very grateful. And then through that, in part, Dan Santi, who was a UCSF faculty member at the time in Pharm Chem, got in touch with us while still at Rockefeller University. Dan, in one of his many ventures, wanted to discover drugs as much as possible through computation. And so he brought together experts in protein structure prediction, or what he thought were experts in protein structure prediction, and experts in virtual ligand screening to fund prospect genomics. So he included people listed here on the left, mostly at UCSF. You'll recognize the names. And the basic idea was to overcome limitations of modeling and virtual screening by looking for golden nuggets among many attempts to model and dock. So even if most of the times you fail, if you can recognize a few times you don't fail among a large number of attempts, which you can do because you have the right infrastructure, you may be generating something useful. And so the technology that we were developing for automated large-scale modeling was appropriate for this task, and we benefited, courtesy of Dan. And without this connection, that then basically established I wouldn't be here perhaps at UCSF, and I almost certainly would not be involved substantively in the other companies as indicated here later. The next spin-off of automating and developing comparative modeling was really a segue into integrative structure modeling. It served as a bridge, this effort for integrative structure determination of large complexes. We realized that uncertainties in the information from template structures used in comparative modeling and uncertainties in molecular modeling resulting from using cryo-EM maps of limited resolution, especially at the time, were often comparable in magnitude but distinct in nature. And so we simply we developed various modeling protocols that combined the two types of information, considered alternative templates, alternative alignments, and alternative conformations in the context of cryo-EM maps when modeling a single or multiple subunits in a larger protein system to improve the accuracy of the models, output models. And one of the applications of this approach came out of a collaboration with Günter Blubell and Joachim Frank in 2001. It was the first complete eukaryotic ribosomal structure, following the first ribosomal structure from prokaryotes just a year earlier from others. And I then met David Eggert at a Gordon conference in 2002, somewhere in the East Coast. And so he described his newly founded QB3 Institute and the forthcoming Mission Bay campus. And as you can imagine, the QB3 vision was really music to my ears. And so I moved from Rockefeller to UCSF. And of course, Kathy, who was my first department chair, made everything happen in practice as only she can. I have to acknowledge them. And one of the lessons here is that it's okay not to be too afraid to throwing chips up in the air every so often and change things and see how they resettle. So at UCSF, even though we still continued to develop comparative modeling just a little bit for a few years, we did focus on integrative modeling since 2003. And as Jamie already mentioned, the first major set of publications from our flagship project, that on the nuclear pore complex, came out in 2007. But from the beginning, in 2000, 2001, this project up to this day and going forward has been underpinned by Mike, Rout, and Brian Chait at Rockefeller, and here and there other scientists, of course, also as well. So the nuclear pore complex on the left is the gate to the nucleus, the blue ring sitting in the nuclear envelope in green. It spans the double membrane of the nuclear envelope and mediates macromolecular transport between the nucleus and the cytoplasm. And obviously, the evolution of the NPCs must have been a key step in the evolution of eukaryotes. The problem for us was how to determine the structure of the system. And we wanted to study, of course, evolution, function, assembly, disassembly, modulation of function based on that structure. Why there was no structure at the time? Well, the system is large, complex, heterogeneous, not possible to crystallize up to this day. And what was the solution? An integrative approach, of course, as you can imagine, the 4 stages occurring in another incarnation here. This just briefly outlines the major types of input information that we used: stoichiometry, copy numbers from the quantitative immunoblotting at the time, the very coarse shapes, basically spheres or a small string of bits of flexible shape inferred from bioinformatics considerations, ultracentrifugation experiments. And then we knew about the symmetry, shape, gross shape, and size of the system from electron microscopy at about 60, 70 angstrom resolution at the time. And then we knew a little bit about protein localizations in the context of the NPC, from immunoelectron microscopy, and maybe most importantly, we knew about protein-protein proximities from basically affinity purification experiments as indicated there. This information was converted into spatial restraints, and the configurations of components were found through structural sampling that satisfy these restraints as well as possible. And the result was a cluster of solutions indicated by these blobs corresponding to the topology model of the nuclear pore complex based on this information. And it's clear that the only way you can get something that's not a bowl of spaghetti is if you use all of this information together, not just each one on its own. There's a nice publication story here, just real quick. So we did ambitiously submit an article to Nature describing both the approach, including modeling and the model, the output. And we were not sure what would happen. And then they asked us they said, no, you have to split the paper into two, make each one longer, and give us back 2 articles. And it was really nice. And also this time there was a news and news as well, but it was only good without any negative letters following its publication. So that was good. 2 more slides on this, just real quick, highlighting 2 specific aspects of this study. One is underlying the point how obviously more information results in a higher precision model, but this is doing it really concretely and specifically, not in an imaginary fashion. For one particular protein in the NPC, when you apply progressively more data as illustrated at the top, you're reducing the uncertainty of the volume in which the protein which you're localizing is localized according to this model. And you can see you go from this large space encompassing the entire NPC in the nuclear envelope. You don't know where the protein is. As you apply this more and more data as indicated above, you get a pretty precise localization. There's multiple copies in the symmetry that you can see reflected in these positions. And the second point is that model assessment is essential. You always have to assess your model and the data on which the model is based. And we were particularly anxious to do so for the NPC structure because it wasn't that we had much experience doing these sorts of structures that we could use to gauge or titrate our confidence in them. And so we came up with these 5 different ways of increasing, hopefully, it turned out that was the case, our confidence in the model. They were basically all variations. Most of them were variations on the theme of how well does a model match known information about the system, whether that information was used or not used to construct the model. And actually, it was really kind of cool to see some of the giants in the field, like the Roots and Kendrew, when they had to defend their first X-ray structure. Their assessment was basically this particular sentence here, exhortations almost. What are the odds that an incorrect structural model of myoglobin, the atomic structure that they had, would result in computed X-ray diffractions that are so similar to the experimentally observed diffractions as we observed for our model? That was the key to their defense. And so we had kind of variations on the theme. But let me just, it's so nice though, because it's easy to convey. What was the time when I realized that maybe we have something in this journey? I don't know, 2005 or 6 or something like that. This is just 2 panels to show that, I think. There was a totally independent data set about clustering of NAP expression profiles. There's 30 different proteins in the nuclear pore complex. They were expressed in 128 different conditions. And then the expression profiles for these NAPs were clustered, and there's 3 clusters indicated for these 30 NAPs or so, indicated by these 3 colors here. Absolutely nothing to do with our exercise of our modeling. We haven't considered it, we haven't been aware of it until then. And so now what happens if you plot these colors on our model? If our model is bad, there should not be any regular pattern, right, of these 3 colors in the model. And if the pattern is not random, then a reasonable explanation is that our model is not random, it's correct. And this is what happens. You get 3 concentric shells. You can rationalize that on top of it, but that non-randomness was enough to start feeling better in the dark, so to speak, before we fully completed this study. Over the years, we then improved this initial model on the upper left by adding near-atomic subcomplex structures, sub-nanometer resolution structure of the entire yeast NPC, describing multiple forums on the left, and then near-atomic model as opposed to the sub-nanometer model of the entire NPC, and finally other parts of the NPC like the nuclear basket, all static structure models, all based on increasingly more and more very different kinds of information, most recently involving a lot of cryo-EM, cryo-ET, cross-linking, and other mass spec methods, and of course, better methods, bigger computers. We did many other targets as well, target systems, always in collaboration with experimentalists. And this is a stamp collection slide illustrating some of these models. These models are many different kinds. Some are static, some are dynamic, some are representing heterogeneous systems. Some are at high resolution, some are at low resolution. Some are molecular networks. All of them computed by a version of integrity modeling. And out of all of this came an abstraction of the modeling effort, the 4-stage scheme or a version of it that's shown on this slide. We have some input information that has to be converted into an output model, and that is done in 3 steps. Some information may be used to describe what we call model representation. What are the parts of the system that we are going to move around in solving the structure modeling problem, atoms, larger units, proteins, domains, whatever the case may be. Another part of information is may be converted, or is generally converted into a scoring function, into special restraints that contributes to a scoring function. And in the 3rd step, at least optionally, some remaining information may be used in limiting the searching for good scoring models. And then importantly, we may not get it right the first time around. And so we actually need to iterate through this scheme until we establish harmony among the pieces of a model, between the model and the data, and among the data themselves, based on various considerations, including the tests on the left. Does the model satisfy all information used to compute it? Does it satisfy information not used to compute it? And is it sufficiently precise to be useful for what we wish to address with that model? I was going to say a little bit more about the scoring function. Let me try, but this may be a little even more chaotic. The scoring function is really the key in integrity modeling because it incorporates a lot of input information that's used to produce the model. It's the heart of modeling, often at least. The purpose of a score is to quantify how consistent is a model with the input information or some subset of it. And as best as I can tell, currently, the best scoring function is the Bayesian posterior model density, which we construct using Bayesian inference, which is why this approach to modeling is a version of Bayesian inference. And a Bayesian inference is fundamental to how science works. It seems that our brains have evolved to apply Bayesian inference, not machine learning, Bayesian inference. And when we can do so, we feel we quote unquote understand things. And Bayesian inference is based, this is a little simplified, but it's based on a very simple starting point. The probability of 2 independent random events A and B is, or even if they're not independent, they can be expressed according to this expression. I'm sure you're all familiar with it. We all went through that in high school at the latest. And it's just after a little bit of rearrangement, we get the base expression here, the posterior probability model density, dependent on two different kinds of information, D and I, it's just labels, is proportional to what's called the likelihood and a prior that have the form is indicated here. And that's all there is to it. You do have to think, or I had to think a bit over time and get familiar with it and so on. But once you do that, it's quite powerful and simple. And a few more comments to maybe make it slightly more connect you with it a little bit more. Of course, the overall probability model density, posterior model density, dependent on all information of two types, D and I, can be factorized into separate likelihoods and priors for the individual features based on subsets of information when they are statistically independent. If you take the log of that, you get the sum of the logs of terms. And that is a generalization of statistical mechanics where the energy of the system is often expressed as the sum over energy terms. And it's also a generalization of the standard traditional least squares scoring functions as indicated down at the bottom. And you're basically replacing, Gaussians with the logs of them, which are harmonic functions that you're so familiar with in statistical mechanics or least squares scoring functions. So I am going to say that anyone who's serious about modeling has to internalize Bayesian inference. It's as fundamental to what we do as time-dependent Schrödinger equation is for science. And here, one more really short vignette. You know what? I will skip it. No, I won't. I won't because I really like it. I'm sorry. I will run out of time. So an important point, which in part can be used to justify integrity modeling, where you really try to maximize the information you have in the input, is that the transition from useless to useful models or from useless to useful modeling can be very sharp. The details matter. And that's illustrated on this plot here. You have the density of states. It's a Gaussian function, very close to it in many, many modeling situations. And when you plot the score or energy, some kind of a score on the x-axis and the number of different models on the y-axis. Obviously, when you have a perfect score, a perfect potential energy function, the correct model, the lowest energy structure, will be in the tail of that Gaussian, will be on the left in this case. But we don't have, in general, a perfect scoring function or a perfect potential energy function because we don't have enough information and other imperfections. And so the actual score we have will score the correct model worse than some of the incorrect models, which are then in this case called decoys. But so that's the setups. Now the very important basic feature of the Gaussian function is that as you increase the fractional accuracy of the score on the x-axis, the surface area decreases exponentially. So you improve the accuracy of your score just a little bit, and you will exponentially reduce the number of decoys that score better than your native state. And so suddenly you will go from a totally useless method to a totally useless score because there's so many incorrect models that score better than the best one you find, to a situation where that's not the case. Exponentially, that will happen overnight. Maybe that's how AlphaFold 1 over zeta. And I think this is something that is generalizable in life, even before modeling. So remember that concept, maybe, because it tells you what to do about it. Pay attention to details, use as much information as you can, because that may push you over the brink more than you'd think. All right, over the years, we've been implementing computational aspects of integrity modeling into an open source program now called IMP. We decided to start from scratch, not continue on Modeler, because we needed to be open source. Modeler was licensed, unfortunately, before I knew better. commercially. We also wanted to make it much more flexible in terms of presentation, scoring, and sampling. It also organizes our group because different people contribute to it. They collaborate with each other to solve bigger problems. And very nicely, it's very nice when it happens. Sometimes students and postdocs move on with the module they developed in IMB, and they continue to develop it in their own research groups. So we then don't have to, we can then do other things, and they're happy, we are happy. And it's really a social way of improving our efficiency as well, this way of organizing our effort. Another example of dissemination is our contributions to a community resource for archiving integrative structure models and the data on which they were based. So in 2014 already, there was a recognition that probably integrative structural biology was important for structural biology. And so we were able to convince the Protein Data Bank, the Worldwide Protein Data Bank, to sponsor a workshop in England to discuss issues relevant to integrative structure modeling, some of which are highlighted in yellow here. And out of that, we wrote a white paper, made a number of recommendations about how to address these issues, and actually followed with the implementation. We became a member, a group at UCSF did, of the American PDB, and through that, we helped construct an actual archive that stores integrative structures and the data on which they're based as part of the PDB. This year, it's being integrated into the PDB proper, so it'll disappear as a distinct entity reaching a major milestone for integrative structure modeling, because it'll become a full citizen of the structural biology nation, so to speak. There were two things that needed to be done in order to achieve this. We needed to cover a large number of different types of structural models, from high resolution to coarse grain, from single state, multiple state, heterogeneous mixtures of models, models that are ordered in time in a network or in a cycle, for example. And we needed to be able to combine all these different attributes, all of which goes beyond the traditional atomic single state, for most part, representation of the PDB so far. The other advance that we needed to implement is organize the resource as a federation of separate archives for structural models, and then the different data on which these models were built, like cryo-EM or cross-linking or FRET spectra or small angle scattering profiles and so on, because it's impossible for any organization, even as large as that of the PDB, to take care of everything. And so we needed to divide and conquer the problem, work with communities generating the data, so they could adapt or create, in some cases, their archives and connect them through APIs to the Protein Data Bank resource with structural models in the center, and then create this scalable, much more scalable structure than would be possible if we wanted to do everything ourselves alone. And then importantly, going forward, we are engaged in assessing, developing better methods for assessing models of any type, including traditional structural models from a Bayesian perspective, and that's for the future. Based on all these experiences, here's something for young people, computational biologists who wish to maximize their impact. It's just a suggestion, consider it. It's good if you are engaging on all fronts that are listed here. Identify, develop, and identify an idea or a concept, implement it into a method, benchmark the method, apply the method, maybe in collaboration with others, and then disseminate it. Do all of the above, not just one task or a subset. Then you will maximize your impact in our experience. So that's the suggestion for you. And I think if you do this sort of thing, then the sky's the limit, and people will want to collaborate with you. And over the years, we have been parts of a number of different consortia that are centers and so on that are listed here many of which are centered at UCSF, but not only at UCSF, and most of them are actually still active. I had some lessons about computers, but I think I will skip that. The next slide shows the first of the two ways in which we're trying to expand the scope of structural biology, as I kind of already alluded, and that's by incorporating the types of experimental information that hasn't been previously used for creating structural depictions of biomolecular systems. In 2008, Nevan Krogan was just a QB3 fellow and basically a glorified postdoc. And so he wanted to give a group meeting to our group, and he did. And he made a proposal, and it was totally his proposal for how to use genetic interactions that he was mapping towards structure characterization of complexes. And it was 12 years later that we published a paper on the topic successfully. The idea was to use the degree of similarity between two proteins in terms of how they respond, their phenotypes of their mutations respond to different genetic environments, such as knockouts. And the hypothesis was if two proteins have perturbations that result in similar responses to knockouts of all sorts of other proteins, then those two perturbations are likely to be close in space compared to two proteins that don't have such perturbations. And statistically, if you do this analysis on known protein complex structures, you can fit, you find that that's true, and you can fit a quantitative expression that characterizes the pattern I just mentioned. And that's a spatial restraint. And you can fit it into IMP, our program for integrated structure modeling, together with known structures or approximate structures of the subunits of complexes, and you can get a reasonable model, structural models of complexes in some cases. And that's an in situ method that Nevan proposed. Doesn't require isolating a sample. In that sense, it's like cryo-ET or in situ cross-linking, for example, but complementary to it. And it's kind of always been very impressive to me that you can go from a petri dish to a structural model of a complex through integrity modeling using this approach, at least in some cases. And then the second type of boundary we try to expand is by developing different kinds of models, structural models, spatiotemporal models is an example that previously haven't been determined based on experimental information. And that's illustrated by constructing a model of the assembly process of the nuclear pore complex. How do the 500 plus proteins come together in the context of the nuclear envelope to form a mature nuclear pore complex? And that's a collaboration with Jan Ellenberg's group at the EMBL. In mammalian cells, you're probably familiar with the cell cycle. On the left, the NPC assembly can occur at 2 points after mitosis during interphase. We study the former process here. The starting point was a schematic, kind of a description of this assembly process, very coarse-grained, based on genetics, microscopy, biochemistry considerations, no calculation. And we simply aim to add as much detail as we could via formal integrative modeling based on some experimental information and other considerations. So again, another form of the four-stage scheme. The key experimental data in this case were shapes of the assembling nuclear pore complex determined for single particles with time-dependent cryo-electron tomography with the time resolution that you see on the slide. We also had, by Jan, of course, the time-dependent copy numbers with some uncertainty for the components of the growing nuclear pore complexes. And then we, of course, knew we had to end the assembly process in the mature NPC architecture we mapped previously. And we had some theoretical considerations as well that I'm not belaboring here. But we simply modeled possible structural models at each time point, and then we connected them using some considerations that eliminated lots of possible pathways through the possible structural states at each time point to obtain a model that satisfied the data, or a set of models that satisfied the data as well as possible. And we validated the model. We used it to rationalize certain depletion experiments and so on. In another example of in integrative structure modeling, solving a problem that you couldn't solve with a single method. Now, of course, over the years, we didn't determine just the static structure of the NPC and how it assembles or models for this aspects of the system. We also proposed how it originated evolutionarily, the NPC. We modeled the transport of macromolecules through the NPC. Barak Raveh, who was a postdoc, followed by coupling the transport through the single nuclear pore complex with a range ATP molecular network in the cell that provides unidirectionality of transport. And we're currently involved with various collaborators on mapping transient interactions with proteins and complexes in the neighborhood of the NPC on the nuclear and the cytosolic side as well. And so the feeling is certainly that in order to understand the NPC maximally and to explain as much as we can about the NPC, we cannot consider these models separately from each other. We really would like to harmonize them with each other. We'd like them to be consistent with each other as much as possible. We'd like to, in a special case, would be using the static structure of the mature pore as the endpoint of the assembly pathway, but we'd like to do that formally, systematically, objectively across all of these models to the degree we can. We are developing a different kind of modeling, we call meta-modeling, to do just that, to integrate different types of models with each other. And just to underline this point, let me state it again slightly differently. In integrative structure modeling or in integrative modeling, we're combining different kinds of data, pri-EM, chemical cross-linking, to get an integrative structure model, in this case, of the ribosome. In meta-modeling, we are combining and harmonizing different types of models with each other. So an example of that would be combining a model of the ribosomal structure with a model of a molecular network that may include, say, some of the ribosomal proteins or its partners. These are two different kinds of a problem. The first one is a characteristic of the type of modeling that's done within a domain of science, structural biology or systems biology. The second one, combining different types of models, seems to be necessary for bridging formally gaps between different domains of science, such as structural biology and systems biology. And so I think the latter, meta-modeling, combining different models, is very interesting, if you can do it. And I'm guessing it will be very important for cell biology, and especially for the impact of structural biology and cell biology going forward. And so we started to address this as concretely as we could. And the first proof of principle wasn't actually a nuclear pore complex, but it was glucose-stimulated insulin secretion in pancreatic beta cells, inspired by Ray Stevens and the Pancreatic Beta Cell Consortium that we are a part of. This definitely is a multi-scale system. There's obviously tissues, cells, pathways in cells, molecules in pathways. And there's models, separate models for each one of these aspects of different size and time scales. And 8 of such models, 2 of them are just data tables, are illustrated here. And we developed a process we call Bayesian meta-modeling that proceeds through 3 steps, I guess, that takes the input models in their original raw form, as other people publish them for most part, converts them into a common probabilistic language called probabilistic graphical models of a certain kind, and then couples them by thinking right now, not particularly systematically, objectively, or automatically, but couples each model to at least one other model, imposing another probabilistic graphical model on top of it, and then synchronizing, harmonizing these models with each other, adjusting them, considering their uncertainties, to make them statistically as consistent with each other as possible through a process called backpropagation. And if we do that, you can imagine, if you can do it, I'm not saying we can, but if you can do it, there's a number of advantages. Well, first of all, I should say that in practice, we certainly need to continue developing this. There's a lack of objectivity, automation, optimality, as I described the process, or not describe it as it currently exists. But in principle, there's all these advantages. And let me just, if you can do this sort of thing I described well, and let me illustrate just one of them, because there's a practical example to make it a little more believable from the proof of principle. Contextualization of two models with respect to each other. Have two models that have nothing to do with each other. They're separate. They may have some variables, X and Y, that they describe their behavior or their values separately, independently. You don't know, though, how X depends on Y. But if you can merge these two models with each other, if you can make them dependent on each other through meta modeling, then you will know how X depends on Y and how Y depends on X in that meta model. And that's a huge gain in principle in understanding and explanatory power that the model can now, the meta model can now provide. That's what I mean by contextualization. An example of that, an actual example, positive example, is our reproduction of the incretin effect, which is a complicated relationship, empirically observed, not really mechanistically explained, about the relationship between GLP-1 concentration as a function of time and insulin and glucose levels as a function of time after meal ingestion. And you can throw in agonists as well that regulate GLP-1R and impact on these relationships. And in our meta model, these are the 4 input models out of the 8 that we used. These variables occur separately. None of the input models have them all together in one, but a meta model does by construction, as I indicated there. And if you then plot the relationship between these variables from a meta model and compare it with the empirically observed relationships for the incretin effect, you get qualitatively perfect agreement. So that's nice. We breached the scales. We provided a more complete and explanatory model in a meta model than we had in the input models. So if you can do this sort of thing reproducibly and well, and that would be very valuable. I will cut a few slides now. I was going to make some comments about current and future interests to do with combining statistical modeling of the kind I described with machine learning. That's non-trivial, but combining these two major paradigms to convert input information into output models seems a worthwhile exercise, doing it non-trivially, that's promising. In particular, that could result in a solution of a major problem in modeling that's still not solved, and that's how to choose an optimal model representation. And I just don't have the time to do that. But I will show you the last slide. Just assume that we are a little bit successful with what I was talking over the last few slides. And let's dream a little bit for this last slide that I will share, make some comments now on it. Imagine that we have a natural resource, doesn't exist, but imagine that we had a resource that can take that has access to all the community databases of all data and all models that different people generate for different aspects of, say, the cell or some complex biological system. And then this can be imported into this cell lab resource and fed into meta modeling process, where the modeling scheme for combining different types of models updates these models in view of the data to make them as consistent with each other as possible, using some criteria I was guessing on the previous slide to obviate the manual intervention that in practice we are still faced with. And then after this update is achieved, the models are validated and again distributed, and meta models included, distributed to the community for the next cycle of experimentation and exploration by everyone. That's basically implementing in slightly more detail that cycle of science consistent with experiments and modeling that we started with. And in this way, again, we might be able to increase more harmony between different aspects of the model that's generated, or a meta model that's generated, between the meta model and the data on which the meta model or models are based, and between the data themselves. And that's how we started. And that's also how we came to from the beginning to the end. And in the end, I'd like to again thank all the collaborators. I'm sure I missed some people, but the mentors I highlighted, there's a few I didn't mention, but the ones are in green. I highlighted the co-editors of a journal, the co-founders of companies. That makes a difference too. And then lots of collaborators during my PhD and postdoc days, and especially now, and look at how many are there at UCSF. And then I just didn't find a way. You see, I ran out of time. I couldn't mention the red ones here explicitly in terms of research, but they know why I'm super grateful to them, and I at least wanted to highlight them this way. And then finally, here are the students and postdocs that went through our group, the current ones on the left. And I mentioned again only a subset. I couldn't mention everyone. And then over the years, there's more. And one of the things I realized, and that I'll leave you with that, is recently actually, is part of that conference whose symbol is indicated on the lower left, that some students and postdocs organized, ex-students and postdocs organized in India this summer. I realized that it's not the papers that are the main output of us scientists, our papers, but it's educating young scientists, these people here. That scales much better if you want than papers. So if you want to maximize your impact, as a scientist, make sure that you have an impact on young people who then go on and do their own science. And I was just in awe listening to the talks at that conference in India, in Pune, India, of all places, where one after the other for 2 days, 2.5 days, they just went and gave great talks about things, many of which had nothing to do with what we do in our lab, but some of them did. And that's kind of the bottom line. And thank you for your patience and attention.
So on behalf of the Academic Senate Committee on Research, we're delighted to present you with the Certificate of Award for your outstanding achievement in basic science research.
Thank you very much.
And we have a little under 10 minutes for questions, so we'll go back and forth between the room and Zoom. Yeah. Maybe we'll take the first question.
Andre, that was a wonderful talk. I wonder when you think about it, fantastic talk. Yes, it is. That was a great talk. Thank you so much for that. Amazing arc of your career. And I wondered, in thinking about putting together, a model of a large complex thing like the nuclear pore, which of the data pieces was the most difficult to get? Like, I could imagine that something that sounds simple, but it may be tough, is stoichiometry of the parts might be hard to get. But maybe you can comment.
Yeah, it's not an easy question, in part because the value of a certain piece of information is not, for structure determination, is not absolute. It depends, it's contextual. It depends on what else you know. So it becomes a very complicated balancing act when you try to say what's more important than the rest. But it's probably fair to say that if you omit, for the initial 2007 study, if you take out any of those major rectangles of information that I had, everything would unravel. We needed each one of them. We did such experiments, computational experiments. I think the affinity purification data, I'll just say that's a little vignette. we used only 80 or so affinity purifications from the labs of Mike Rout and Brian Chide, very focused experiments, high quality control. And that allowed us to compute models that existed in 3D and satisfied the data. If we took affinity purifications from databases generated by lots of different people, maybe with a little less care on the average, maybe not necessarily every contributor, like Nevin Krogan never makes mistakes, of course. But if you just take data from a large public database, you can't calculate a model that satisfies all the data. There are more mistakes than you think. You would have to assume a big uncertainty, not assume, you have to assign a big uncertainty to the data, and then maybe there would not be enough information to get a usefully precise model, even though, with big enough uncertainty in input data, you could satisfy it, obviously. So there's games like that we play all the time. We never succeed, almost never succeed the first time around, right? We convert the data into representation, scoring and sampling, do that, go through that process and either don't get models that satisfy all information, or get models that are all over the place, or get models that satisfy most of the information, but really violate something they shouldn't violate. And we have to troubleshoot it. We have to go back and change how we see information, how we convert it into any one of the three aspects: representation, scoring, and sampling, until we're happy. And there's no method at the moment anywhere in science that I'm aware of that can do that automatically. My last 3 slides were about a potential solution for that. I didn't get to talk about them. But automating that in an objective and optimal fashion is, I think, if I had to pick a single problem in modeling for biology, cell biology, when you're trying to address the cell modeling problem, mapping problem, is probably one of the key, if not the key bottleneck on the modeling side. So I squeezed in comments on the last 3 slides after all. David.
Yeah, as Jim said, absolutely fabulous, Andrej. If I understand it right, I mean, for the meta modeling, which is a fabulous concept, am I right in thinking that in each of the individual modeling experiments that you're trying to then combine, that there have to be effectively latent variables, or whatever you want to call it, of the other model context that is present for them to be synergistic and give rise to a kind of a greater whole?
You have multiple choice. You can think about it that way. At the moment, you have multiple. We see as one having multiple choices as to how you couple each input model to at least one other model. One way is via physics. Another way, let me give you a concrete example in the case of GSIS modeling with insulin. Insulin occurs in many different incarnations in these different models. It's not exactly the same variable, but it's a similar one. There's insulin concentration in the cell, there's insulin concentration in the plasma. There's insulin inside the granules inside the cell. There's insulin that's synthesized, right? And so on. There's insulin, it's a different insulin. But if you think about it, and that's the problem, you have to think about it, and it's manual at this point. You can couple, at least statistically, these different insulin variables in different models to each other. That's what we did. But you could do it, there's maybe other ways. You could do it with physics, if you had it, protein docking, flexible.
If I could, right?
Yes,
I mean, I'm trying to get at, that for you to be able to couple models, there has to be some kind of intrinsic information which is being shared. It may not have been explicitly varied in the context of one set of experiments compared to another. But how do you identify that? I mean, because if you're going to have a zillion models that you're now trying to merge in different ways, you have to be able to then ask, is the synthesis greater than the individual ones themselves? And so how do you answer that or approach that in a general way?
No, if I understood you correctly, again, you're absolutely correct. If you are faced with coupling 2 models that are so far apart, that you can't connect them in any reasonable fashion. I don't know, a model of an atom and a model of a galaxy, and you know nothing about the physics that connects the two. It doesn't matter how much meta-modeling you do, you're not going to connect these two models. So but you can ameliorate this problem if you have more models, so that in the input, because you only need to connect each model to at least one other model before you benefit from the coupling, if you can do it. You don't know it. You don't know it. And in fact, you may be able to find out that there isn't any need to change the input model, a given input model, in order to make it more consistent with any other model, right? I emphasize the context insights that you may gain from meta-modeling, where you suddenly relate a variable from one model to another variable in another model, where you had no clue that the two things were related, or how they were related, even less so, right? So that's contextualization. But you may also find, by way of explaining a bit more about meta-modeling, relevance to your question, you may also find that given the presumed input uncertainties of the input models, you cannot adjust them enough to make them statistically sufficiently consistent, which means possibly you over-interpreted the input models, and now you have a chance to go back and do something about it, probably do more experiments, but you are guided in that effort. Or you may find, there's certain variables that are just not coupled with each other according to meta-modeling, corresponding to your question. So all these different outcomes are possible. And in fact, we did search systematically for pairs of variables, each one from a different input model, that are maximally correlated in the meta-model. That's how we found the incretin effect. So it really, you have to trust me, but it's like it's a perfect story of the potential power of combining different models with each other, as opposed to different data only.
Question on Zoom now.
Hi, so we have one question on Zoom. It might be similar to what we were just talking about, but Sergey Kotelnikov asks, hi, Andrej. Is it possible to reach integrative modeling harmony with any combination of experiments, or sometimes the experiments might be incompatible with each other, different conditions, environments, et cetera, and trying to combine data from these experiments might frustrate integrative modeling?
No, I like this question. So, the answer, there's two answers, conceptual and practical. The conceptual answer is absolutely yes. In the way to think about the problem that was included in the question is exactly through, I suggest, through our framework for statistical inference for integrative modeling. Because the problem in that case that you'd be addressing with the aid of our framework is to find an optimal representation. Should you explain the heterogeneous data sets from, say, a crystal and solution for one protein, in which, and in the two samples, they may assume different states. What you need is a two-state model. You need a combination of two states together in one model, where one state explains one set of conditions and the other state explains the other set of conditions. And we have projects that do exactly that. I think the first example was with Bill DeGrado in practice. We had a functional cycle, two states, equilibrium simultaneously in the membrane. There were crosslinks on these two states coming from a mixture of the two states. Any single state, as best as you could compute, could only explain about half of the cysteine disulfide bridges. He had 200 of them. But if we ask the question, what's a combination of two states that best explains the set, we could explain almost 100% of these disulfide bridges. And so it's a matter of looking for the representation, for the set of variables, for the type of model that that can explain the data, all the data sufficiently well. That's the harmony that I was referring to. And you cannot do it completely automatically. You cannot do it automatically right now. And that's the final set of remarks referred in part to that. But you can do it manually with the tools we have and often solve it in practice.
Hi, Andre. Great talk. So I have a bit of a personal question. The way you tell your story of your life in science, going from high school until giving this award talk to the UCSF faculty, your interest is primarily in comparative and integrative modeling, and then you're using Bayesian inference to accomplish that. Out of all the computational methods, how did you decide to focus on Bayesian statistics and modeling?
I didn't decide, actually. No, I really think we all evolved, whether we know it or not, to think in a Bayesian fashion, some of us more cryptically than others. And you know, even if you talk to some serious machine learning people, they will say, you know, we suffer from not quote unquote understanding machine learning. Machine learning is a black box. There's a lot of effort to make it transparent, quote unquote. But but that is achieved by making it more Bayesian inference-like. And that's great because we want to do that. One of the things I didn't mention, or maybe I did, is we want to combine machine learning paradigm with statistical inference paradigm in a non-trivial fashion. But I think the human brain evolved to think in a Bayesian fashion and to be happy feeling that it understands things when you can explain what's going on in a Bayesian fashion. And some of us do that more intuitively than others. In the modeler paper, when I go back, you know, I even showed you the equation. It's it's an approximation of a Bayesian formulation. Even smoothing the sparse data, because I had 7 structure alignments in the late 80s to infer those spatial restraints from, or the tables for them. Treating the sparseness was done in a Bayesian fashion, but I had no idea that it was Bayesian. I certainly didn't call it that. Just later, it turned out to be the case. It's innate in us. I'm not talking about myself. I think it's a human thing. You know, what's more natural? Yeah, I wish I could explain it better. So one problem with this talk is you only give it once. There's no chance to practice it much. So you got a very raw deal. I'm sorry, the first one and the only one.
Thank you. Thank you, Dr. Sali, for that wonderful talk, and thank you to all of you for attending this event. Please nominate your colleagues for the faculty research lectures. Nominations are open now. We have basic science, clinical science, and social behavioral science. And for those of you who are here, please join us in the lobby for a reception. Thank you so much.